Good day all. We, Team B1, present you our e-poster on the topic types of vaccine. Prevention is always better than cure. One such prevention is vaccine. A vaccine is a biological preparation that provides active acquired immunity to a particular infectious disease. A vaccine typically contains an agent that resembles a disease-causing microbe and is often made from weakened or killed forms of the microbe, its toxin, or one of its surface protein. Vaccines don't just work on individual level, they protect the entire population. It provides herd immunity.
In regard to all this information, a national immunization schedule has been created taking into account the more fatal diseases as well as those which are endemic to a particular area. To present you with a short summary of the national immunization schedule in India, the vaccines given at birth are the BCG, OPV or oral polio vaccine 0 dose, hepatitis B birth dose. At 6 weeks, the first doses of OPV, pentavalent vaccine, rotavirus vaccine, and PCV vaccine are given, pneumococcal conjugate vaccine. The subsequent doses of these vaccines are given at 10 weeks and 14 weeks. Between 9 to 12 months, the MMR vaccine is given, and between 16 to 24 months, the booster dose per MMR as well as the DPT vaccine.
Come, we'll go there.
Toxoid vaccines use a toxin made by the microorganism which can cause a disease, and they kill only the toxin instead of the whole microorganism. The mechanism of action is that toxoids are weakened form of the toxin, so they cannot induce any clinical features in humans which the disease actually causes. They are more stable and they are less susceptible to damage which is caused by temperature, humidity, or light. An example of toxoid vaccine is the DPT toxoid vaccine. It is a trivalent vaccine which has diphtheria, pertussis, and tetanus. It contains toxoids of only Corynebacterium diphtheriae and Clostridium tetani. Variations of the DPT vaccine are TDAP and TDAP.
Vaccination schedule is complete. But is there any other vaccine that you recommend taking?
Since it's the flu season, you might as well get your flu vaccine done.
I'm a little concerned. Is it actually necessary?
See, it's not necessary, but since flu is very common in India, it's better to stay safe and get your vaccine done.
Inactivated vaccines are made from viruses or bacteria that have been killed or inactivated, rendering them unable to cause disease. They can also be used in immunocompromised patients. However, they require multiple doses to achieve optimal protection. Hepatitis inactivated vaccine is stored at 2 to 8 degrees centigrade. It is given intramuscularly in 2 doses, but it is not a part of the national vaccination schedule. Inactivated polio vaccine provides low levels of immunity locally in the gut. It provides protection against paralysis but not against spread of the virus. Rabies vaccine can be categorized into tissue culture and embryonated egg origin. Standard schedule is 0, 3, 7, 14 and 30 days since exposure. Influenza vaccine is recommended in India for high-risk individuals. Currently, it is not a part of the national vaccination protocol. Edible vaccines are subunit vaccines where the selected genes are introduced into the plants and the transgenic plant is then induced to manufacture the encoded protein.
Viral vector vaccines are the liquid or freeze-dried preparations containing live viruses that exhibit low or non-pathogenicity towards the target species. So these viral vector vaccines basically use viral vectors to deliver the genetic material, DNA. Vaccinia virus and adenoviruses are the most commonly used viral vector vaccines. The advantages of viral vector vaccines is that they have a better presentation of antigen and also generate T cell response. The disadvantages of them are that they can cause bad reactions. They often can only accommodate 1 to 2 antigens.
Live attenuated vaccines use a weakened form of the germ that causes the disease. They help to prevent and create a strong, long-lasting immune response. For example, MMR and varicella vaccine. While viruses attenuated by repeated passing in cell cultures for more than 10 years transforms into the vaccine virus. Small doses of this live attenuated vaccine is injected into the host. Replication of this vaccine virus inside the host produces a specific immune response. Induction of an immune response gives protection against the real infection.
Varicella vaccine. Chickenpox is a highly contagious viral infection which causes an itchy blister-like rash on the skin. This vaccine is given in 2 doses. The first dose is between 12 and 15 months and the second dose is between 4 and 6 years. There are 2 types of vaccine, Varivax and Proquad.
MMR vaccine. MMR vaccine protects against 3 diseases: measles, mumps, and rubella. This vaccine is given in 2 doses, the first dose between 12 to 15 months and the second dose between 4 to 6 years. There are 2 types, MMR2 and Priorix.
Subunit vaccines contain only a specific antigen or protein of the virus along with an adjuvant to give better immune response. Example, SARS-CoV vaccine, HPV vaccine. Moving on to a type of subunit vaccine, recombinant vaccines, produced by genetic engineering techniques. As we all know, a gene of specific antigens of the pathogen after inserting into a harmless host is purified and then is used as a vaccine. These are easy to develop and are more safe. Now let's clear a major dilemma between our next 2 vaccines, the conjugate and polysaccharide vaccine. The polysaccharide vaccines elicit immune response to the body by using the polysaccharide molecules to cover the bacteria, induce type-specific antigens, and thereby enhance opsonization, phagocytosis, and the killing of the bacteria. Whereas the conjugate vaccines is a step ahead as it addresses the limitations of pure polysaccharide vaccine as it combines the sugar unit with a carrier protein that facilitate better activation of immune cells, bringing out effective immune response against the pathogen when required.
mRNA vaccines. mRNA vaccines work by introducing a piece of mRNA that corresponds to a viral protein, which the cell can use to produce the viral protein. The immune system recognizes the protein as foreign and produces antibodies. In case of future exposure to the virus, antibodies recognize and destroy it.
Advantages. mRNA vaccines have proved to be very effective against infectious disease targets in animal models of influenza, Zika, and rabies virus. They have also been used efficiently in cancer trials. One of their biggest advantages is their non-infectious platform and that their in vivo half-life can be regulated through modifications. These modifications also make mRNA more stable and highly translatable.
Disadvantages. The properties of mRNA vaccines and their interactions with lipid nanoparticles make them unstable, which reduces their global accessibility. For example, COVID vaccine produced by Pfizer needs to be stored below -70 degrees Celsius.